A very warm good morning to all of you from PSGVP Mandal, SI Patil Arts, GB Patel Science, and STKVS Commerce College Shahada, District Nandurbar, Maharashtra. Again, you are welcome for the workshop on techniques in plant anatomy organized by Department of Botany under DBT Star College scheme. I am extremely happy to share with you that we have more than 1500 registration with international participation.
नहीं डबल नहीं
I
लागला
welcome
एका
all
एकच आहे.
delegates, researchers, all students for our workshop.
पाहिजे
Now, I request our Vice Principal and IQAC Coordinator, Dr. M.K. Patel, to introduce our esteemed institute.
Good afternoon, everyone, and a warm welcome to all. I, Dr. M.K. Patel, Vice Principal and IQAC Coordinator, take this opportunity to welcome each one of you to the inauguration of the DBT Star College scheme, which is sponsoring this workshop on techniques in plant anatomy, which is organized by Department of Botany, Pujya Sane Guruji Vidya Prasarak Mandal's Arts, Commerce and Science College Shahada. I extend a warm welcome to Honorable Professor Dr. R.S. Patil sir, who is the Dean, Faculty of Science and Technology, Kawetri Bahinabai Chaudhary, North Maharashtra University, Jalgaon, who is chairing this session. Honorable Professor Jay Panda sir, who is from PP Institute of Science, MKP University, Bhavnagar, Gujarat. Coordinator of the DBT scheme, Professor R.Z. Syed sir. Professor S.K. Taide sir, who is HOD of the Botany Department. Convener, Professor N.K. Athole sir. Organizing Secretary, Professor M.B. Jagtap sir. Advisory Committee members, our Vice Principal, Dr. S.D. Sinketkar sir. Organizing Committee members who have helped to grace this occasion, members of the Botany Department, and my dear participant students who have joined from all over the country to grace this occasion. Friends, we are proud to host this event on behalf of the management of Pujya Sane Guruji Vidya Prasarak Mandal Shahada, and I welcome you all for this one-day e-workshop. Today, we have with us a distinguished and a learned guest, Professor Jay Pandya sir, who is associated in the field of life sciences, as well as he is a active and diligent researcher who will enlighten on the topic of anatomy. Friends, we are indeed much fortunate to be with you today, as our institution is celebrating the Golden Jubilee year, and as one of the prime initiative of this process, today we have organized this webinar. Our college has been constantly encouraging our faculty and students to develop a system by organizing such type of workshops, seminars, conferences for all the stakeholders for the catalytic action to improve our academic as well as the administrative performance of our institution. All the teachers, they are trying their level best to reach to the students by some other means, because we are not able to conduct our sessions physically. So for maintaining the quality initiative, its sustenance and enhancement, we need the participation of all the stakeholders, the management, administrative staff, students, parents, employers, and so on. From March 2020, we are all facing the worst ever pandemic, that is COVID-19. But still, under such conditions, we are constantly feeling that some concrete academic activities should be organized for the benefit of our students. And in this link, today, we are continuing our efforts. Uh, During the last 1.5 years, we have been organizing many national conferences, workshops, and other activities for our students. And the aim of organizing such type of workshop in botany was initiated by our HOD Professor Tide sir and his team members. Active participation of the students is must. So as rightly expressed by Professor Atole, we have been celebrating this grand event by participants of 1,500 plus and so on. So this affirms the relevance and meaningfulness of this event. So this is the real synergy of youth and creativity and the talent which is expected in this such type of seminars. So I am very happy to organize such activity in our institution that will really boost our confidence in future. We will always say that we have added members to our family and we are all professionals now and we are growing as a family member. So such type of programs for all our resource person will help us to grow together. Friends, you can learn today more things, much things from this workshop because all of you are learned persons and you will be trying to commenting and asking questions at the end of the session also. So we really honor the spirit of learning from you and we wish to keep this spirit alive in future also. So this is a very good opportunity to network with each other. So after this workshop also, we urge that you should network with our family members, with our botany department, with our resource persons, and then you try to work interdisciplinary work in the future also. So at this juncture, I will express that we will together try to learn and grow with the participants as well as with the resource persons. We are forced to go for online, and we have no other choice now. And to the bright side, so much participants have added today. So this will really help us to boost our confidence. So with this short note, I thank Dr. Pandya sir for accepting our invitation. And in future also, if some activities are conducted physically, we will definitely call you, sir, and we'll take the services of your expertise. Thank you, sir. Thank you very much.
Thank you very much, sir.
Thank you very much, sir. Now I request our principal Professor Dr. R.S. Patil, sir, to chair today's workshop.
Thank
Also, it's my privilege to forward our botany department, which is established in 1970. Sir, it is one of the famous department of North Maharashtra University, and many renowned teachers have worked here, which is now working in other universities like Gujarat University and all that. It is recognized research lab under various PhD students are working. Also, well-equipped labs are there, and botany department offer PhD and post-graduation with the specialization angiosperm taxonomy. And we provide various facilities for graduate and postgraduate students. In the last year, we have 42 students in the TYBSc level. Also, we have carried out various research projects, many publications of all the faculties are there. So, we are organizing this STEAM workshop under the botany department. Now, I invite the DBT Star sponsored scheme coordinator, Dr. Sayed sir, to share his talk. Sayed sir.
Thank you, Dr. Atole, for giving me this opportunity. President of this e-workshop, Honorable Principal Professor Dr. R.S. Patil, sir, who is also Dean, Faculty of Science and Technology of KBC North Maharashtra University, Jalgaon, Vice Principal and IQC Coordinator Dr. M.K. Patel, Vice Principal of Arts Faculty Professor Shinkhedkar, Head Department of Botany Professor Tade, Convener of this workshop Dr. Atole, Organizing Secretary M.B. Jagtap, other colleagues in the Botany Department, and participants, coordinators of DBT scheme, from the various colleges in the country, and dear students. A very good morning to one and all. First of all, I take this opportunity to congratulate the Department of Botany for organizing such an important workshop on the plant anatomy. I welcome you all in the capacity of coordinator of this DBT star scheme of this college. Professor Athole has mentioned that there are 1,500 participations from various states as well as from abroad. This overwhelming response is definitely a result of dedicated efforts and teamwork of botany department. I congratulate to them. Friends, this DBT star scheme has been launched by Department of Biotechnology, Government of India, and this scheme is aimed to brand and nurture the excellence in UG science education. This program particularly emphasizes the holistic approach for the improvement of science education at undergraduate level with the special emphasis on practical training to the students. This scheme is meant for providing the knowledge and skill of teachers in the basic sciences and specialized techniques. This scheme also provides access to the specialized infrastructure to the students, assurance of consumables, substantial hands-on experience in designing and conducting new additional practicals, and it also gives opportunity to assess books and journals. So these facilities which are otherwise not possible without the support of DBT have been provided by this DBT star scheme uh, by Department of Biotechnology, Government of India. Department of Botany has organized one workshop earlier. This is their second workshop. Under this scheme, Department of Zoology has also organized one workshop. Department of Biotechnology has organized two workshops. One was on the intellectual property right, and another one was on the bio entrepreneurship. And there are many such programs. There are also some hands-on training which will be conducting after the arrival of students in the college. So these all programs are meant for the improvement of students, for providing them the additional facilities. And I request all the students to take the benefits of these various workshops, seminars, summer schools, winter schools, and hands-on training. Lastly, I take this opportunity to the organizers to thank the organizer for giving me this opportunity. Thank you very much.
Thank you, Syed sir. Now I request our head of the department, Professor Dr. S.K. Tayade sir, for introduction of our esteemed guest, speaker Dr. Jay Pandya.
Thank you, Professor Athole. Good morning, one and all. Honorable Professor R.S. Patil, Principal, Pujya Sane Guruji Vidyaprasak Mandals, Arts, Science and Commerce College, Shahada. Honorable Dr. M.K. Patel, Vice Principal and Coordinator, IPSC. Honorable Dr. A.D. Sinkalkar, Sir, Vice Principal. Honorable Dr. R.Z. Syed, Coordinator, DBT Star. Honorable Dr. J.B. Pandya, eminent speaker for this webinar. Convener, Professor N.K. Athole, Sir. Organizing Secretary, M.B. Jagtap. Dignitaries, teachers, researchers, and students. I welcome you all in this e-webinar entitled Techniques in Plant Anatomy, and it is my great pleasure to introduce today's eminent speaker, Professor Dr. J.B. Pandya. Presently working as a assistant professor at Sir P.P. Institute of Science, M.K. Bhavnagar University, Bhavnagar. He has completed his BSc in Botany from Sardar Patel University in 2007 and then MSc in Botany from Sardar Patel University in 2009. Similarly, he has completed his research work as a PhD from M.K. Bhavnagar University in 2020. He has 10 years of teaching experience. He has received 2 national awards. First is the first prize for the best poster at the National Science Symposium on Drainage, Waste, Solid and Water Management, organized by Dist College, Saurashtra University, Rajkot on 6 January 2008. And the second one is Best Poster Award at a national seminar on fundamental science, present global scenario and future perspective, organized by Sir P.P. Institute of Science, M.K. Bhavnagar University, Bhavnagar on 1st March 2020. He has 8 years of research experience and he guided for 3 MSc students for their project and dissertation work and published 19 research papers in national and international reputed journals. He has also authored 2 books and participated in 32 national, international conference, seminar, and workshops. Thank you. So, over to
Thank you, sir.
convener, Professor.
Yes, thank you, sir. Now I
Thank you.
invite our principal, Professor Dr. R.S. Patil, sir, for presidential address.
Thank you, Professor Athavale. Good morning, one and all. Patrons of this national workshop on plant anatomy, Honorable Bapu Sahib Deepak Bhai Patil, President of Pujya Sani Guruji Vidya Prasarak Mandal Shahada, Co-Patron Honorable Kamal Tai Sahib Patil, Secretary Pujya Sani Guruji Vidya Prasarak Mandal Shahada, and Honorable M.N. Patil Sahib, Coordinator of Pujya Sani Guruji Vidya Prasarak Mandal Shahada, Resource Person Honorable Dr. Jay Panda, Dignitaries Online, Vice Principal and IQC Coordinator Dr. M.K. Patel, Vice Principal Dr. S.D. Sinkadkar, Head of the Botany Department Professor S.K. Taide, DBT Coordinator Professor R.J. Sayyed, Convener of this workshop Professor Nilesh Athavale, Organizing Secretary Professor M.B. Jagtap, Professor Vijaya Patil Madam, Professors and students, greetings of the day. Hope you are doing well in this pandemic caused by COVID-19. Friends, I take this opportunity to introduce our college in brief. 50 years ago, a renowned personality, freedom fighter, Sahakar Marshi, and foresighted visionary, late Sri Anna Sahib P.K. Patil, initiated noble cause of education in the form of Pujya Sani Guruji Vidya Prasarak Mandal Shahada. Pujya Sani Guruji Vidya Prasarak Mandals, Art, S.I. Patil Arts, G.P. Patil Science, and STKVS Commerce College, Shahada laid its foundation in 1970. The college has a progressive management led by Honorable Sri Bapu Sahib Deepak Bhai Patil, President of our PSGVP Mandal Shahada. It is a multi-faculty, co-educational, government-aided college affiliated to Kavaitri Bahinabai Chaudhary North Maharashtra University, Jalgaon. Our college is has a permanent affiliation of the affiliating university. Friends, the college has been felicitated with the College with Potential for Excellence status confirmed by UGC New Delhi, and the college has been awarded Star College status by Department of Biotechnology, New Delhi. And this program is organized under the DBT Star College team. Friends, our college is a URSA beneficiary college, and our college is assessed and re-accredited by NAC Bangalore in 2017 with A grade and CGPA 3.12. Proud we feel to mention our position A grade college, as ranked by the affiliating university, that is Kavitri Bahinabai Chaudhary North Maharashtra University, since last 5 years. The educational history for more than 5 decades and healthy traditions of the college attract students not only from the state of Maharashtra but the state nearby states like Madhya Pradesh and Gujarat also. The faculty comprises of more than 60 learned members and experts in their own subjects, offering their valuable services with a sense of dedication and devotion. The college, after laying its stepping stone in 1970, has been recognized by UGC New Delhi under 2F and 12B scheme of the UGC Act 25 and has a permanent affiliation, as said earlier, with the affiliating university, KBC in Jalgaon. With the introduction of graduation courses in basic subjects, we have now reached the present status wherein 16 courses are offered at UG level, 13 courses are offered at postgraduate level, and our 11 departments are recognized as a research center by the affiliating university. The emphasizing aspect worth mentioning is the faculty facility for acquiring PhD degree in 11 subjects. Our 11 departments are recognized departments, recognized research centers by the affiliating university, and the students are working under the valuable guidance of more than 20 eminent supervisors, with the rarely observable at which is rarely observable at a college level. The motivation from our academic coordinator, Honorable Sri Hemant Patil sir, have played a pivotal role in propagating quality initiatives in the college. Friends, as we know that plant anatomy or phytonomy is the general term of study of internal structure of the plants. Originally, it includes plant morphology, description of the physical form and external structure of the plant. Today, renowned personality Dr. Jay Pandya from Surat will speak on the plant anatomy and its techniques in this national workshop, that is a national eWorkshop. Our team of botany department in collaboration with IQAC under the DBT Star College scheme have put lot of efforts for the organization of this workshop. I am sure that this workshop will be helpful for the students of life sciences in general and students of botany in particular. My best wishes for the success of this workshop. Thank you. Jai Hind, Jai Maharashtra.
Thank you, sir. Now I request and invite our speaker, Dr. Jay Pandya, to enrich us with his knowledge of anatomy. Welcome, sir.
Thank you very much, all the authorities and principals and Dr. N.K. Athwade sir, Dr. M.K. Patel sir, Dr. N.K. S.K. Taide sir, R.S. Patil sir, and M.B. Jagtap sir for inviting me for such a nice topic. And herewith I am sharing my screen with the blessings of Puja Guruji, Sane Guruji. I will start my today's lecture. My slide is visible now.
Yes, sir. Yes, sir. It is visible.
Okay, thank you. The topic for today's lecture is techniques in plant anatomy. Plant anatomy is a very vast subject. We cannot complete it within a hour time or in a single session, but still I will try to incorporate as such as detail possible, which will be beneficial to the all the students of BSc and MSc. My target is mainly specifically for the BSc and MSc student of botany, so they can have an interest in plant anatomy and they will work very good in plant anatomy. Now, micro techniques. The word micro techniques, it means the sections can be prepared for a microscopic examination and observations. The aim of that study to examine the cells and tissues under the microscope, how the plant's structure is arranged, its architecture, and etc. So we can know the structure of that particular plant, and we can use it for the identification purpose also. Yes, we can identify the plants morphologically also, but anatomical evidences are also needed to make the surety that it is particular plant. So plant microtechniques is a very much essential technique to identify and to know the structure of that particular plant. There are many methods available for the microscopical observations, such as smearing, squashing, maceration, hole mounting, clearing the sections, and sectioning and staining. And one by one, I will explain all the techniques with possible examples. This is a smearing. We are preparing the smears for the bacterial stains and fungal staining, or for the pollen culture. So this is a technique of smearing. By this, we can prepare a smear on a slide, and we can spread the liquid form or semi-solid form or a solid form on a slide, and then we can stain it and observe under the microscope. The first this slide is gram staining method for the bacterial staining from the curd, gram positive and negative bacteria. And the second slide is a pollen of a Catharanthus rosis. For the identification of pollens, we are preparing the slide by a smearing method. That is the advantage of the smearing method, that we do not have to go for the dehydration, embedding, and sectioning method. It is very easy technique. So we can prepare the slide by the using the smearing method. This is a procedure. First, we have to cut or take the sample of the anther, and with the help of scalpel, we have to placing the pollens on a glass slide, and then we have to squeeze it, and tissues are then it will come out, and we have to spread it uniformly on a slide. And then we have to remove the anther, and we have to stain it with a appropriate stain, such as acetocarmine, aniline blue, cotton blue, phlogiston stain, or Bismarck blue. These are the common stain which are used for the pollen stain. The next method is a squash preparation. The squash preparation we are mainly using for the study the mitosis and meiosis in a plant tissue. Mitosis we can observe in onion root tips, and that meiosis we are observing in a anther or bud of the flowers. So in a squash preparation, it is a very fast technique and very easy to prepare the specimens. And for that, we have to take the sufficiently thin and transparent material to view under microscope. The procedure for the squash preparation is we have to place a drop of water in the center of the slide, and then put the small piece of the material in a water. We have to carefully place the cover slip on the top of the specimens, and we have to crush it or squash it, and then we have to go for the staining procedure. So onion root tips or petals or tendrils or many other things we can make with the help of squash technique. The staining or procedure for the squash method is acid hydrolysis. With the help of 10% HCl, we can make the material easy for the acid hydrolysis, and then we have to remove the stain, remove the acid portion, and we have to stain it with the acetocarmine. The second method is enzyme hydrolysis. And enzyme hydrolysis is more recent technique for mostly in the enzymes are used to digest the plant material, such as cellulose and lipase enzyme. We can hydrolyze with the help of enzyme hydrolysis technique. And then after we have to go for the preparation of the slides, and with the help of 90% alcohol, we are removing the excess amount of enzymes from the cover slip, and then we have to stain the material and fix it with the help of methyl benzol or a drop of DPX mounting media, and we have to fix the material. So to study the meiosis and mitosis, we are going for the process of squash preparation, either with the help of acid hydrolysis or with the enzyme hydrolysis. The next technique is a maceration technique. The maceration technique is, just a moment. Wait a minute. Is it visible now? Hello? Am I audible clear? Am I audible clear? Hello? Just a moment. Now, from the maceration technique, am I audible clear?
Yes, sir.
Yes,
Yes,
sir,
sir.
yes.
Continue. Please continue.
Please continue.
Okay, okay,
There was some network issue.
okay, okay. This is a maceration technique. We are dissolving the, we are doing the maceration in plant and animal material through the hydrolysis process. And we can separate out all the cells or tissue with the helping of dissolving the middle lamella with the help of Kuzel method or Jeffries method or Heron method. These are the type of maceration. Fluids method. In this technique, the material is treated with the concentrated nitric acid and a few crystals of potassium chloride. When the material are completely bleached and it is washed with the water, and then we are staining the material and look into the microscope. The same thing is happen with the Japanese method, but treatment is different. The material is treated with the 10% of chromic acid and 10% of nitric acid for 30 to 45 degrees Celsius for 1 or 2 days. We have to keep the material, and then after we have to wash it thoroughly. The material are separate out and macerated well, and then we have to prepare the slide with the help of proper staining method. And these two methods are, we can uh, prepare the temporary slide, but the third method, hollow method, is for the permanent slide preparation. For the maceration slide, the material are treated with the chlorine water for 2 hours. After the wash it with the running tap water, we have to, and the material we have to put in the boiling sodium sulfide for 15 minutes for the maceration. The material are hydrolyzed and bleached, and then we have to wash with the silver. Then we have to stain with the safranin and remove the excess stain with the help of distilled water. And when it is dried properly, we have to fix it with the Canada balsam and we have to observe it in a microscope. So by the preparation of first and second method, we can prepare the temporary slide. By the help of hollow method, we can prepare the maceration slide in a permanent slide. This is a method for hole mounting. In some cases, we have to observe the reproductive structure of a particular plant, or we have to mount the embryo, and at that time this method is very helpful. This is a mounting of sexual organs, globule and nucleus from a cara algae, that is freshwater algae. In whole mount method, we have to preserve the material and either in from a stem, leaves, or flowers, or fruits, or reproductive organ of any lower plants. We can prepare the whole mount slide with the help of preserving it. Some problem will be happen that we are not able to retain the natural color or form or shape of that particular uh, plant or material. But if we use a proper fixative, and we can use a proper clearing agent, we can maintain that. The microscopy observations are prepared by the use of ethyl alcohol or formaldehyde. If we can use a 2% formaldehyde in 70% alcohol, that is best material for the algae and bryophytes and pteridophytes to make a whole mount slide. And according to material, we have to modify the protocol for the preservations. Now, tissue clearing. It is very much essential to clear the tissue for the observation of epidermal or exodermal or endodermal layers. For the leaf, if we want to observe the stomata, it is very much essential to clear the tissue. And for that, in this technique, for the sectioning of living plant tissue, we have to use fast microscopic observation for the internal structure. The best thing to use appropriate clearing agent. If we want to mount the means if we want to observe the stomata from a monocot leaf, just we have to keep the plant material in a 5% formaldehyde to 10% formaldehyde for 15 to 20 minutes, or depending on the material, if we kept for 1 hour, the epidermal layer will detach from the mesophyll layers, and we can very much easily observe the entire structure of uh, stomata and epidermal layers or appendages or etc. So it is for the wall mount also we can use it, or for the studying the exo and endodermal layers. The clearing of the material is very much essential. Now the preparation of permanent slide or temporary slide or semi-permanent slide. Okay, so in the case of this is the first figure, that is temporary slide preparation, and the second photograph shows the permanent slide preparation. What is temporary slide? We have the face material of stem or leaf or any plant material. We can prepare it in a temporary slide preparation or semi permanent slide preparation with the help of having a hand sections. For the permanent slide preparation, we have to go for the sectioning with the either with the hand microtome or rotary microtome or sliding microtome with the proper micron sections. So now the temporary slide. How we can prepare the temporary slides? The study of the transverse section of stem or root of dicot or monocots in from plant material, we can prepare the temporary slide. The common mounting media for that is distilled water or glycerin, or that is sufficient to prepare the temporary slide. We have to use with the sharp razor blade and brush and needle. With the help of watch glass, we can prepare the temporary slides, and we can observe the sections. The procedure is described over here, and we all are using in laboratory, we are taking rapidly temporary slides. Okay, so now the semi-permanent slide. This is not done in every laboratory, but it is very much essential to teach the preparation of semi-permanent slide. That is very much essential. Semi-permanent microscopic slide prepared in done by the two different mediums such as Carocon medium and glycerin jelly medium. What is Carocon medium? It is after the satisfactory fixation with the 3 to 5% formalin, we have to transfer the specimens into watch glass or on a slide. And at the point of material may be suction with the necessary, the stain with the specimens with a solution approximately 1% alanine blue or other stains as a acid stain, erythrostain or safranin. We have to uh, go on taking the sections and fix it with a Carocon medium. Then we have to wash it with a 1% HCl and a few drops of distilled water. We have to use it as a naturalized medium, and then we have to add the Carocon medium on diluted in a distilled water, and with the help of that we have to make the mounting media, and we have to fix the slide in a Carocon medium. Next is glycerin jelly preparation method. For that, we have to use gelatin, glycerin, and water. We have to mix it in proper amount and heat it on either in hot plate or in on a burner. And we have to prepare the slide. And instead of glycerin, we have to use a glycerin jelly to fix the material. By using these two methods, we can prepare the semi-permanent slide, which we can store it for 2 to 5 years. So it is beneficial to prepare a semi-permanent slide instead of a temporary slide. So this is the protocol of the application of the glycerin jelly slide. Now towards the permanent microscopic slide preparation. So in a permanent microscopic slide preparation, by using the clearing method, killing method, fixation and hardening method, sectioning method, then we have to follow the staining, dehydration, again clearing, and mounting and labeling. These are the steps to prepare the permanent slide. And for that, what is clearing? The tissue must be cleared for the study of an internal structure of cell or tissue. We have to place that particular material in a 10% of potassium hydroxide or sodium hydroxide solution for overnight. After initial clearing, place this material in an acidic medium, and we have to neutralize that to the alkaline condition. The second step is killing. Killing or fixing the material, it is very much essential. In this first step of preparation of permanent slide, it is very much too important. By killing the specimens, we can stop all the activities of the cell in a specimen. So we can get the specimens in respective original state. The maintaining original state, it is very much essential to study the histochemistry or biochemistry of that particular cell. This can be done with the help of using 1% osmic acid and ether. Fixing and hardening process. The fixing and hardening is the second major step to prepare the slide. In case of a minute living organism, it is usually attained by the killing agent alone. But in case of sections of having larger organisms or tissues, it may be done through the various fixating agents. The process of fixing has multifarious importance. Fixing to stop any alternation of the form of tissue. It will harden the tissue and makes them fit for extensive subsequent treatment. It makes the tissue suitable and successive for the action of stains and various other reagents. The fixing agents make various constituents and components of a tissue optically differentiated by the changing their refractive indexes. These are some fixative agents, that is Bowen's fluid, mercury chloride, 70% alcohol, acetic acid, formalin, potassium dichromate, or osmic acid. Depending on the which kind of material we are using to fix, we have to use the appropriate fixative agents. After the fixing of any of the affordable fixative, we have to wash the material. That is very much essential. Otherwise, tissue will be completely spoiled. So, whatever fixative we use, the appropriate clearing agent or washing material is there. For 70% means if we have used the points fixative, we have to wash it with the 70% alcohol. For mercury chloride, we have to wash it with iodine plus 70% alcohol. For acetic acid, we have to wash it with the 50% alcohol. For formaldehyde, we have to wash it with the 70% alcohol. For chloral hydrate K2CrO7, we have to wash it with the water and 12% chloral hydrate. Or for osmic acid, we have to wash it with the water. So whatever fixative material we have used, if we are not washing it, there is a no use of preparing a permanent slide. In many cases, I have seen that people are not going to use appropriate that washing chemical. If we have stain it with Bowens fluids, and if we are washing with water, there is no use. We have to wash it with a 70% alcohol only. If we have used acetic acid, we have to wash it with the 50% alcohol only. So appropriate washing is very much essential. Otherwise, tissue will be completely spoiled. And when we come to know that tissues are getting damaged at the time of staining, when the entire process is completed, we have prepared the slide. At that time, we came to know that tissues are getting damaged. Okay, so following all the steps, it is very much essential. Now the sectioning. How we are taking sections? Yes, traditional method is taking hand section. In BSc and MSc laboratory, we are taking hand sections or with the help of hand microtome only. Okay, so having a appropriate angle for the hand sectioning, it is very much essential. We have to maintain the 45-degree angle in between the plant material and our sharp razor blade. Then and then only we can have a good material. The sections are thin, it is parallel, and it is not going oblique. We have to take the care for that. But in hand microtome, the that angle is already given, and we can take the hand section by using the sharp razor blade. The first one is rotary microtome and second one is sliding microtome. For the sectioning in rotary microtome, we have to pass the material through the processing for the wax embedding sectioning. We have to embed the material in a wax, so we can have a ribbon of serial section. For the serial sectioning, we have to use the rotary microtome, and the entire processor we have to pass it from a TBA to the para TBA, and then in a paraffin, and then after paraffin we have to prepare a block, and by the trimming and proper block with the help of microtome blade, we can take a section, and then only we can prepare a ribbon for a serial section of that particular plant material. For nodal anatomy, we are using such kind of serial sectioning, or the fruit material we are using the serial sectioning. But for the harder tissue, it is when the tissue are already harder, we are taking the section on a sliding microtome. For a wood anatomy especially, we are using the sliding microtome. Staining. After the having a proper sectioning, staining is a very much essential to develop a contrast or to stain a particular tissue with the help of proper stain. The acidic stains are there, neutral stains are there, basic stains are there. So whatever material we have to stain, we have to use the appropriate stain. And for that, different staining techniques are there. Either single stain, we are using one stain to stain the cytoplasm and nucleus, that is single stain technique. But if we want to go for the specific kind of staining, we have to go for a double staining method or triple staining method, or n number of stains we are using at a time. So staining is a very much important technique. uh, we are going for a in this session we are going for a special kind of staining techniques in detail later on. Then after dehydration technique, after the staining we have to prepare the slide for a permanent slide. So removal of water from the tissue is very much essential. For that we have to pass the entire slide from a dehydration series. For that, we have to pass it from the 20% alcohol, 30% alcohol, 50% alcohol, 70%, 90% in pure alcohol. Uh, uh, in a gradually, when we pass through the uh, 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 gradual series, the water amount will be removed. If we keep the slide directly in a 100% alcohol, the sudden loss of water cannot be possible, and the tissue will getting shrink. So it will be damaging our slide and tissue at that time. To achieve a proper dehydration from material, we have to use the proper timing and proper gradual increase to alcohol series. After single or double staining, we have to remove the excess stain by washing the tissue with acid alcohol, and then after we have to go for the clearing or de-alcoholization. That is very much essential because the mounting media will not bound with the alcohol or acid alcohol. So clearing or de-alcoholization is very much essential. For that, the suitable dehydrating agent, alcohol or salicyls, whatever we have used, we have to clear it with the clearing agent. And generally, we are using the cedar wood oil or clove oil, or most commonly, we are using that clearing agent is a xylene. For that, we have to mix up the xylene in a proper ratio, either 3 gem 1 or 1 gem 1 or 3 gem 1. And then after ratio of alcohol, initially the alcohol is more and xylene is less. Then one in 1 gem 1 both are in equal amount, and in 3 gem 1 the alcohol amount is less and xylene amount is more. We have to pass it gradually, and then after we have to give the 2 changes of pure xylene to remove all the amount of alcohol. That is very much essential. And after the alcohol, we have to use the proper mounting media to fix that slide, to prepare that it permanent. And for that, we can use the Canada balsam dissolved in xylene, or Euroferol or DPX. Commonly, we are using the DPX or Canada balsam dissolved in xylene to prepare a permanent slide. Euroferol we are using sometime only for a particular slide preparation, but DPX is a very much cost effective also, and it is very much transparent. So we are using DPX as a mounting media for the preparation of permanent slide. When the slides are stained or dehydrolyzed, and at that time after the clearing all the amount of alcohol, when it passed through the pure xylene, after that with the help of glass rod, we are putting the drop of DPX on a section, and then we are placing the cover slip on that, and we are allowed it to dry. After 1 or 2 day, the slide will become permanent. This is very small thing, but very much essential. Labeling, proper labeling should be there for each and every slide. For that, whatever the specimens number we have mentioned, we have to mention that. Whatever the plant material name, from where we have found, from at what date we have prepared the slide, or a collection date of that slide, or any other detail, whatever it is, a LS of so and so, or TS or TLS, RLS, whatever kind of section is there, we have to mention on the that particular label. So any one of them uh, easily can recognize what they will observe in that particular slide. If the proper labeling is not there, if the section of maybe we can say it is a leaflet of cycles or reaches of cycles, but it is ls or it is a ts, or how much old that slide is prepared. So we have to mention all the details. Who has prepared that particular slide? Yes, we can mention that. So labeling is that much essential after the preparation of permanent slide. If we not label it properly, uh, if we have 4 or 5 slide, yes, we can easily find out what is our requirement. But after having 100 slide, we are not able to find it easily what slide we are going to use. We have to check every time. Okay, so arranging in a slide rack, it is very much essential. Or identification for the identification or reusing the permanent slide, it is very much essential to label it properly. So in the both age of that particular slide, we have to make them labeled. Now the stains, different staining techniques, either single stain or double stain or differential stain and special staining techniques. We have to use it. I must say we have to incorporate all these staining methods in our at least MSc syllabus. So students will have the benefit of that, and they have the interest in plant anatomy to by using this kind of different staining techniques. So a single stain, generally safranin or fast green, we are using alone for a staining purpose. If we want to uh, stain the algae, we are using the fast green or safranin, or for the bryophytes or some pteridophytes or spore of a pteridophytes or pollen grains of gymnosperm or angiosperm, we are using aniline blue or safranin, whatever it is suitable. With the help of single stain, we are staining entire plant material. That is safranin or fast grain or aniline blue, whatever we are using. What is the procedure? Okay, we have to take. We are taking a small amount of stain in a watch glass, keeping the material in a stain, and again washing it with a proper washing material, and preparing a slide with the help of mounting media either as a water or glycerin. That is single staining method. Whatever in BSc and MSc we are doing, that is single staining technique. Okay, so in that case, the first photograph shows the preparation of single staining method. The another photograph is showing the double staining method. This is safranin fast grain stained material. We are observing that the primary tissues are stained with the fast grain, and secondary tissue or complex tissues are stained with the safranin. So two different stain we are using at a time for staining a single slide. That method is a single stain technique. Sorry, double stain technique. So, how to prepare a double staining technique for temporary or semi-permanent slides? So we have to take the sections, we have to select the good sections and stain it with the safranin up to 4 to 5 minutes. We have to wash it with the water. Again, this stain, if the stain is more, still we have to wash it with acid or alcohol or whatever is necessary. We have to again wash it water rapidly and stain with the pass it to the alcohol series or and stain it with the fast grain whatever, because in double staining we have prepared the fast grain in a alcohol, so we have to pass it through the alcohol series and up to 90% 95% alcohol, we have to take it, and after that we have to stain it with a fast green or aniline blue, whatever stain we want to use, and we have to wash it again with the alcohol and glycerin and mounting it in a glycerin. So this is the procedure for the double staining technique for temporary or semi-permanent slide preparation. If we want to prepare the permanent slide of that double staining, we have to take the good sections with the help of a rotary microtome or sliding microtome or hand sections, whatever we can take. We have to stain with aqueous safranin or crystal violet. We have to wash it with uh, water to remove the excess amount of staining. Then we have to pass it through the dehydration series: 30% alcohol, 50% alcohol, 70% alcohol, 90% alcohol. Then whatever stain we want to use, we have to prepare that stain in alcohol only. As a fast green, we are preparing it as a 95% alcohol and clove oil. So we have to stain it with a fast green or erythricin, and then after we have to distain it again with the 90% alcohol, then absolute alcohol. Then we have to pass it through the clearing series and dehydration series. So for the clearing or dealcoholization, we are using xylene, and we are passing through the xylene and mounting it in a either in Canada balsam or DPX. So this is a protocol of the permanent slide preparation. So if we want to use temporary or semi permanent slide, the protocol is totally different. If we want to prepare a permanent slide, that is also different. Then this is a differential stain. In differential stain, either we can use the toluidine blue or hematoxylin safranin. What will happen? Tissue having a different kind of biochemistry or phenomenon. By the applying a single stain, two different kind of tissue is stained with a different color. In this, we are observing that first is hematoxylin or desafranin. Well, we have to make a mixture of hematoxylin and safranin. We have to ripen the hematoxylin first, and then after we have to make the combination of the hematoxylin and safranin in a single bottle, and then we are applying it in a leaf material. Some tissues are in stained with a pink material, and some are stained with a blue. So it is a differential stain. With the applying a single stain, we can stain it as a multiple. So it is a depend on the nature of the cell or tissue or stain. Depending on that, the differential stain will be happen. The another slide shows the xylem and phloem structure. So xylem in this second slide stained with the toluidine blue, xylem is stained in a blue color, that is lignified tissue, and non-lignified tissues are stained with the help of purple or pink color. So by applying a single stain, we can easily observe where the lignified tissues are present, where the non-lignified tissues are present. So applying a differential stain, we can easily differentiate it which kind of tissues are present over there. So this is a differential stain. It is very much interesting. In MSc, at least we have to go for a, yes, we are going in MSc, we are going for double staining also. But nowadays we have to incorporate that in a syllabus. A differential staining is very much essential. Now, some special stains. Why I mention over here special stains? Yes, now maybe for the BSc and MSc student, this staining techniques are not known for the students. Yes, as a faculty, we all know that we can stain the some minerals, we can stain some carbohydrates, some protein molecules inside the cell, some lipid molecules, polyphenols. We can stain the lignified tissue or lignin. We can stain the nucleic acid and histones also. But in MSc, if we incorporate these details in a syllabus, it is very much essential to have a development of research attitude in a plant anatomy for the basic botany students. Yes, we are going for a DNA isolation, RNA isolation, or mRNA technique, or RT-PCR technique. All these techniques are very well known, but these are the also interesting techniques, special staining techniques for the minerals, carbohydrates, protein, lipids, polyphenols, lignin, nucleic acid, and histones. So I will go for the some example of minerals. First is aluminium. Okay, for that chrome azurol as method, it is described by Kuchka and Miller in 1980. It is very much old technique, but still it is lab. And these are the photograph of aluminum. In first series, aluminum is absent, and if we see the second series and third series, it is stained in a uh, blue color or orange color. That is, aluminum is present, and this is a difference of a staining method. Okay, so if we want to where the aluminum is located in particular tissue, yes, for that the specific stain is chrome azure z. And if we apply that on a plant tissue, we can found in which area particular aluminum is active, or we can find the aluminum in that particular tissue. Boron, yes, we all know boron deficiency is there, and we are talking about so much about boron for the fruits. If the boron deficiency is there, uh, the fruits are not in a good shape, and it will decay soon, because at the fruit ripening boron is very much essential. But is a particular amount of boron in present in our material? How we can confirm that? Okay, by looking into morphology. After having a symptoms only, we can find out, yes, boron is in less amount. But by the crystal formation method described by Marsh and Shiv in 1941, if we take the section and stain it with a particular stain, the yellowish cells will appear the presence of proper amount of boron in a plant tissue. So if we have some sample and we have taken the sections and stained with the particular method for histochemical localization of boron, uh, before deficiency, we can confirm that the amount of boron is sufficiently present or not present. So we can supply it as a supplement, and we can came out from the after development of the fruits development, or we can minimize the loss of the fruits. Now the calcium is very much essential to develop the salmon. For that, different techniques are there. Silver method is there, alkaline pyrogallon method is there, and silver hydrogen peroxide method is also there. And different scientists have uh, demonstrated different technique to localize the amount of calcium. If we want to locate the calcium oxalate crystal, we have to stain it with the silver hydrogen peroxide method. If we want to see the deposition of calcium in a cell wall, we have to use the silver method. If we want to stain it with a yellowish brown color, the amount of calcium and the area of calcium where it is present in that particular cell or a tissue, we have to use it, stain it with the alkaline pyrogallon method. So this is a result of the histochemical localization of the calcium. Okay, the first slide shows the amount crystals of the uh, calcium oxalate crystals, and second slide shows the amount of calcium that is localized in a alpine galanga. Okay, by the help of alkaline pyrogallon method, the golden color is appear. Okay, in that yellowish brown or golden color will be appear. So it is very much interesting where the calcium is there, how it is look like, in which zone of the tissue, either it is present in a epidermal layer or a cortex layer or a pith region, where the calcium amount is present or calcium oxalate crystals are present. So we can stain it and we can observe them. Magnesium, by the conalization and Titan method described by Borda, we can histochemically localize the amount of magnesium. Magnesium is very much essential mineral for the photosynthesis also, because in the chlorophyll structure, the magnesium ring will be there. So, in a tissue where the particular amount of magnesium is essential, it is a macromolecule as a mineral. So, the amount of magnesium where it is located in a particular sample. Yes, if we stain the different materials, yes, these are the all the these all slides are prepared from a tomato plant. Okay, so this is first one is a stigma, and this is a leaf blade, and this is the developing leaf. This is a fruit material, developing ovary, and these all structures are stained with the quinolizine and titin method. And the blue color, brick red and blue color, will shows the amount of the uh, magnesium present in the cell. Cellulose method for the carbohydrate. If we want to locate the carbohydrates, cellulose, it's a common material. Cellulose is present in a plant cell wall. Okay, so if we want to see the amount of cellulose, how much amount of cellulose is present, or where it is present, or different kind of tissues, some having a more amount of cellulose, some cells having a less amount of cellulose. So for that, chlorazole black E method is, it is described by Robert and Purvis in 1964, and if we stain it properly, we can use uh, develop contrast by staining the same tissue with the uh, chlorazole black E method. Okay, this is most common method. Yes, we are doing this. Starch, iodine and potassium iodide reaction. Starch will appear blue or black in a few minutes, but after completely means when the time passes on, the reaction will be in a reverse phase. So in that particular thing, iodine and potassium iodide, we have to prepare it for a fresh, and we have to take the photograph. If it is required, we have to take it immediately. When the time passes on, the reaction will be reverse and it is decolorized. The very interesting thing in iodine and potassium iodide reaction, yes, we all know the starch will be appear blue or black in few minutes, but if it is newly formed starch, it is appear red to purple color. If we stain the leaf material with the iodine and potassium iodide, that result will be appear. Or if we stain the phloem tissue with the iodine and potassium iodide, yes, we will definitely have the newly formed starch over there, and it will be appear red to purple in color. So something is interesting. We have to develop the interest in this. Now, callose deposition. Yes, we all know the callose is deposited in the phloem sieve tubes. If we use the lacmoid blue as a staining material, callose will stain in a greenish blue color. And these all photograph shows the deposition of callose in a phloem sieve tubes. So the entire section will be in a blue color, and wherever the only callose, particular presence of callose is there, if we have stained the phloem with the uh, another stain, it will be completely blue. But if we have used the lacomoid blue method, uh, it will be appear as a greenish blue, and that is particular the deposition of a callose in sieve tube. So we can see in A photograph the amount of callose deposition is high, but in B sieve tubes the callose deposition is low. While in C photograph, the phloem tissues are completely deposited with a callose deposition. Maybe it is a sealed phloem. It is non-functional phloem. So if we want to functionality of a phloem, we can stain it with a lacomoid blue method. While in D, yes, very less amount of a callose deposition occurs in a phloem sieve tubes. So for all these things, some particular interesting staining method is there, that is specialized stain. We have to use all this method. Insoluble polysaccharides method. Why it is insoluble? Yes, it is insoluble in water. Some polysaccharides are soluble in water, some polysaccharides are insoluble in water. So if we use Parse reaction, periodic acid and skiff reagent, two different uh, chemicals are there. First one is periodic acid, second one is skiff reagent. When the sections are taken, first it is pass it through the periodic acid. We have to wash it with the water to neutralize it. Again we have to stain it with a skiff reagent, and then in a skiff reagent, uh, it is uh, when it pass through the skiff reagent, it will develop a purple or pink color. Okay, the amount of presence of insoluble polysaccharide in a cell, it is visible. Either it is in a purple color or pink color or red color. If we see in this, yes, it is a fruit of Dolichos labdab with a seed. Yes, that this outer thing is a pod wall. Okay, and the inner, that is oblong structure, it is seed. In seed we are looking over here, yes, it is red color, while all other uh, area it is purple or a pink. Or we can see some cells having a dense deposition of a purple, development of a purple color, and some cells have a less development of a purple color. So whatever area having a dense color of purple color, the amount of insoluble polysaccharides are higher over there, and having a less deposition of a purple color, having a less amount of insoluble polysaccharides. So in such a way we can identify that where we can found a more amount of insoluble polysaccharides in a particular plant or tissue or a fruit. So, and in which layers it is abundant, and we can use that. Next is sulfated and carboxylated polysaccharides. First one is insoluble polysaccharide. Next one is a sulfated and carboxylated polysaccharides. So for that we have to use the toluene blue O method, described by the O'Brien et al. And McCulley and Feder and Wolf. In that sulfated polysaccharide or carboxylated polysaccharides, when we applied the toluene blue O, first one we have used, that is toluene blue, and this one is a toluene blue O. So these sulfated polysaccharides or carboxylated polysaccharides, when we applied the stain, they will develop pink to the reddish pink color. Actually the toluene blue have a uh, development of blue color, but if it is stained with the pink to reddish pink, that is sulfated and carboxylated polysaccharide. So if we want to use that particular amount where sulfated and carboxylated polysaccharides are present, we have to use the toluene blue O method. Staining methods for lignin. The most common method is Moll's reactions. The persons who are working with the lignin deposition, yes, that is very much common method, Moll's reaction. For that we have to stain it with different chemicals, and the red color, it is developed with the help of moles reactions. The other method are fluoroglycinol method or toluene blue method that will appear different colors. This is a reaction of the uh, staining of a moles reaction. The lignin is deposited, and where the lignin is deposited, if we want to stain the lignified cell wall, yes, we have to use a moles reagent. Okay, if we want to use the primary cell wall, secondary cell wall, then if we stain with the fluoroglycinol HCl method, we can differentiate it the uh, secondary wall layers. Primary, secondary, tertiary wall deposition will be happen in lignified walls. So we can stain with the fluoroglycinol method. So with the help of different kind of methods, we can identify the layers of the lignin. Then method of staining for the polyphenols. Again, the toluene blue O, that is, if the polyphenols are present, it is develop Turkish green to blue color, or if we use a fast green, it will develop a red to dark brown color that indicate the presence of polyphenols. Next, the tanning. For the tannin method, we are using 2,4-dimethoxybenzaldehyde reagent that is described by Mack and Holle in 1974, and the presence of tannin is appears purplish or red coloration. Or if we are using the ferric sulfate or chloride method, it is appear blue or bluish green precipitate. It is shows the presence of tannin. So these are the presence of polyphenols. Okay, and these are the stains for the polyphenols. If we have used the fast blue BB, it will appear as a different. This is a localization of a presence of tannin. If it is tannin in a hard form, yes, it will appear simply. But if it is not in a hard form, yes, the tannin is present in high amount in different kind of fruits or cells or tissues. So if we stain it with a different stain, either in ferric chloride method or DMB method, it will shows the presence. The first slide is from the root tissue, and second one is a rhizome of a alpine galena. Okay, so in a cotton root or in a alpine galena, that rhizome, we can observe the these are the result of the presence of tannin in that particular tissue. Yes, in some fruit also the tannin is present. So if we are not able to digest a high amount of tannin, if it is less amount, yes, it will not affect us. But if the high amount of tannin is present, we are not able to digest the high amount of tannin. So in fruits, if we stain it properly, so we can identify whether in the especially in underutilized fruits, amount of tannin is high. So we can identify the amount of tannin, or if it is dense, yes, it is having a high amount of tannin. If it is not dense, it is having a less amount of tannin or negligible amount of tannin presence in that particular fruit. Yes, Gorasamli, high amount of tannin present in that particular fruit. But at the time of ripening, yes, the tannin is getting dissolved with the help of some special enzymes, and the amount is decreased. So in which particular stage it is present, we can also locate that. Now for the protein, mercuric bromophenol blue method is there for the proteins, and for that, if it is stained with the mercuric bromophenol blue, the proteins will appear in a blue color. Or the another is dinitrofluorobenzene method. The protein is develop a deep reddish purple color. So with the help of different stain, the reaction will be differ and the final color is totally changed. The most common method for the proteins is amido black P method. It will give us a blue color. So this is a section. These are the sections which are stained with the amido black P, and this all shows a blue color development. Staining methods for nucleic acid and histones. Yes, we can stain the nucleic acid and histone, and we can identify that in a particular plant tissue. If we use the fast grain FCF, it will stain the histones in a green, and it will having a rich green color. So we can stain it with histones with the help of fast grain FCF or naphthol yellow S method or ammonical silver method. These are the different methods to stain the histones. These are the results of the histone staining. Staining of DNA and RNA. Yes, isolation and estimation of DNA or gel electrophoresis of DNA. Yes, all the things are possible. But we forgot to stain the RNA. If we want to stain the DNA or RNA, yes, particular methods are there. Methyl green pyronic method, it is there. In that DNA appears blue and RNA stains also we can identify. If we use a toluidine blue method, yes, RNA stains will convert in a purple while DNA is converted into a blue or blue-green color. Stains for the lipid. Lipids and lipoproteins. For the lipid and lipoproteins, we are using Sudan black tea. Yes, it will appear in a dark color. Okay, it is stained as a black color. And lipid and sterols, we are using benzyl pyrene caffeine method, and that is give us a blue or bluish white fluorescence effect. If we want to use a total lipids, that is most common method is a Nile blue method or oil red O method, that will gives us a dark red color. For lipid histochemical localization, the first one is stained with the Sudan black, and the carpels are stained with the Sudan black, and in the second slide, the pollen grains on stigma stained with the Sudan black. Yes, this is appear in a dark black color. This is the stigma, and these are the pollens. So it is appear in a dark black color. Unsaturated lipids. For that, osmium tetroxide method is most common method, and that will appear in a black color. Wherever unsaturated fatty acids are present, it is appeared in a black color. Okay, now the fatty acid and neutral lipids. For that, the Nile blue A method described by the Ruthman, it is most common method. The first one slide show the osmium tetroxide method stain. The unsaturated lipids are localized with a dark black color, and the second photograph shows the stain with the Nile blue A method, which shows the presence of fatty acids stained with a Nile blue. Okay, so these are the histochemical localization, and these are the very interesting things we have to learn, and we have to teach these methods to the students. Now, phospholipids. For that, acid hematine method is described, and the phospholipid stains in a blue or a bluish black in color. If we are using the orangey or an aniline blue method, the phospholipid stains yellow, and uh, uh, rest part is appear in a blue color. If the unsaturated phospholipids are there, we are using the Otain reaction, osmium tetroxide alpha naphthalene method. And by using this method, the unsaturated phospholipid stains orange red, and cholesterol esters and triglyceride esters are stained in a black color. And this is a photograph shows a phospholipid stain with a orange G. We are clearly see the uh, development of orange color, red color, and light colors to show the presence of this different uh, histochemical localization. Up to this, it is learned by me. But some interesting thing I came across during my research work, but I have not applied in my research because having some kind of limitations. Some histochemistry of enzymes, histochemical localization of particular enzyme. This is a book, Enzyme Histochemistry, a laboratory manual written by Z. Losda R. Gorasarau T. Sisbuller. This is a book. Entire book is uh, described for the histochemical localization of enzymes. These are some reference paper. I clearly mentioned this. I have not done any histochemical localization of enzymes, but whatever I came across just for the development of an interest for the students, I am keeping over here. Plant histochemistry, versatile indiscernible tool to localization, gene expression, enzyme, cytokinin, secondary metabolites, and detection of a plant infection and pollution. This is done by the Farida A. Petria and Vola S. Ebolmati, Department of Pharmacognosy. It is from the Egypt. Okay, so a very interesting thing they have done in 2019. Very recent article is that it is published in Acta Scientific Pharmaceutical Science, a very reputed journal. The next one is histochemical and biochemical studies on some enzymes of Legula intestinalis. Okay, so they have done some work on a histochemical and biochemical localization of some enzymes. Then histochemical localization of cytokinin oxidase and dehydrogenase during the developmental stages of Withania somnifera. Yes, Withania somnifera is a very much important medicinal plant, and this research work is done at the PG and Research Center, Department of Botany of Tamil Nadu, and they have done a very good, interesting work. I have read these all articles, and they are very much interesting. They have done excellent work. Histochemical localization of beta glucourinase activity in plant tissue. Okay, so whatever enzyme we want to do the histochemical localization, yes, it is possible. And for that, particular methods are there. Some precious chemicals are there. We have to use that, and we have to do the histochemical localization of beta galactourinase. This is a most liked or most cited article in situ staining of activities of enzymes involved in carbohydrate metabolism in plant tissue. This research work is done in Netherlands and Russia, and most cited article I have found. The work is done in February 2002 and published in Journal of Experimental Botany, very reputed journal of botany. And this is a very interesting. If someone is interested in that, just go for this. It is free access journal, and very interestingly, it is described, and all the images are shown in that particular tissue, histochemical localization of enzymes involved in carbohydrate metabolism. The next one is effect of salinity stress on biochemical and anatomical characters of ceratophyllum. This one work is done in Iraq. Okay, effect of salinity stress and biochemical and anatomical characterization of ceratophyllum plant. So all the work, whatever advanced work we want to do, yes, we can do, but for the evidence, we have to incorporate the anatomical and histochemical localization of that particular uh, chemical. That will give a very nice combination. The rapid histochemical staining method visualizing the role of fusarium oxypermum catalyze in xylem uh, embolism, formation of lupin wilt. When the disease is formed, whatever the chemical is forming and developing such a to localize that particular chemical in that that is histochemical staining. Okay, so these are the very interesting work I came across to prepare this presentation. So I have kept over there, and whatever I know, I still repeat that I am just a beginner in plant anatomy. I am not an expert in plant anatomy. I am just a beginner or learner in a plant anatomy, and whatever knowledge I have shared it with you, and with the acknowledgement of all my faculty members. From I learned the plant anatomy, I developed the interest of plant anatomy in that, starting from the BSc, MSc, and my research journey and my research guide, Dr. Sailesh K. Mehta. I am very much thankful to all the botany faculty through whom I learn a botany and especially plant anatomy. I mentioned two names, Professor T.V. Raman Rao and Professor K.S. Rao sir, through whom I learned the very good anatomy. I'm thankful to them all. Thank you very much.
Thank you, Dr. Jay Pandya, sir, for delighting us with the knowledge of basic to advanced techniques in the plant anatomy. Now the session is open for the question and answers. So if participants are having any question, they can type in to the comment box, and we'll try to answer it. Please, if participants are having any questions, they can type in a comment box. There was one question earlier asked by some participant. I'll just try to see that. Yes, there was a question from Sudishna PK. Is there any special stain for mercury? She was asking about that. Jay sir, please.
Some fluorescent dyes are there, uh, Fen Green and SK or Fen Green FL. These are the special stains for the mercury. Yes, if you read the plant histochemistry by John S. Haas and Professor K.V. Krishnamurti, they have published the book for the histochemical localization. All the metals are covered in that.
Okay. So, is there any other question? Please post in a comment box. Okay, there is a question from Ramesh Singh ji that sir, what is a tangential section?
Tangential section is the same, the transverse section. Nothing is different.
There is one more question from Chitra Prasad Balakrishnan ma'am. She's asking that how to prepare Carocon medium.
The Corrigan medium, the method I have shown in that particular slide, uh, and we have to prepare the Corrigan medium in dissolving it in a water. So if we search for that, or in plant microtechnics, botanical microtechnics by S.M. Kasim sir book, it is mentioned how to prepare a Corrigan medium. I will show it again. Just a moment.
Yes, sir. Take your time. Make it full screen, sir.
Yeah.
Okay, it's visible now.
Yeah. This is the method. We have to use a 3 to 5% formalin and transfer the specimens in a watch glass, and then we have to prepare the 1% aqueous solution. And the total Corocon medium preparation is given in a botanical microtechniques by SM Kasim.
Yes, any other question, participants? Yeah, I will post in a comment box. Okay, there is one more question from Ramesh Singh. How to condenser in a microscope affect the resolution? Okay, how the condenser in the microscope affect the resolution?
Condenser in microscope affect the resolution because the length of condenser it will vary, and if we don't have the proper condenser, the objective lens and eyepiece will not give us a good result. So condenser for that it is proper condenser is required.
Okay. Okay, so many participants have requested for the PPT. So, with the permission from Dr. Jay, I assure you we will send that PDF copy of that PPT into the Telegram group at the end of this program. And
Yeah,
I have
definitely.
already shared the feedback link with you all, and I will share it in Telegram channel too, Telegram group too. So, please provide your valuable feedback for the program. So, I think there are no more questions from the participants. You can ask your questions in Telegram group also. So
Uh,
we will try to answer it in that group also.
I will provide my email address in a Telegram
Yes,
book,
yes, no
and
problem.
they will make questions over there.
Yes. One more question, sir. Last question. Asha Kadam from Asha Kadam. Please specify two sides of mirror of microscope.
Two sides of my mirror.
Mirror of
One
microscope.
is flat mirror and one is concave mirror. We are using the concave mirror to in a microscope to observe the specimens.
Okay, so further questions will be answered in Telegram group. So you may join.
Yeah,
Already I have shared the link for the Telegram group, and I'm again sending a feedback link to you. Please check your chat box, and I will share it in Telegram also. Now I request Dr. Sunil Patak sir for the vote of thanks. Dr. Sunil Patak sir, please.
Thank you, sir. Good afternoon. Today, it has been a knowledgeable workshop in plant anatomy, which I am very sure that it will be helpful for every attendee. It is such an honor for me to thank all the dignitaries on behalf of Botany Department of PSGVPM ASC College, Shahada. Initially, I thank the organizing committee for giving me this opportunity to give away the vote of thanks. I express my deep gratitude to PSGVP Mandal for granting us permission to organize such a unique and academic event. I give my hearty thanks to our Honorable Chairman Sri Deepak Bhai Patil, Secretary Honorable Srimati Kamlatai Patil, and our Coordinator Principal Honorable Makaranda Bhai Patil. I give my deep salutation to our Honorable Principal Professor Dr. R.S. Patil Sir, Honorable Vice Principal Dr. M.K. Patil Sir, Honorable Vice Principal Dr. S.D. Sinkhedkar Sir, for their valuable guidance and support. A huge gratitude to the esteemed speaker of our today's workshop, respected Dr. Jay Pandya, MKB University, Bhavnagar, Gujarat. I would like to express my gratitude to all delegates of workshops for their presence and contribution to make this workshop a great success. I must thank the organizing committee members, Professor Dr. S.K. Taide sir, Professor Dr. R.Z. Sayyed sir, Professor Dr. I.J. Patil sir, Dr. U.M. Dadhav sir, Dr. R.M. Chaudhary sir, Dr. M.M. Jogi, V.K. Patil, N.K. Athawale, Dr. S.S. Pandey, Dr. Y.H. Vasu, K.N. Koli, Dr. H.P. Jadhav for their efforts to make this workshop successful. I also thankful to our faculty member and non-teaching staff for their support. Thank you very much.
Thank you, sir.
Thank you, sir.